Hi, and welcome to my CME presentation, More Than a Gut Feeling, the microbiome's impact on mental health and cognitive function. Course learning objectives. After viewing this presentation, students will be able to discuss the primary mechanisms of the gut-brain axis, identify key GI hormones and microbes involved in neurotransmission, explain how ultra-processed foods and antibiotics affect the gut microbiome, and analyze the role of probiotics, dietary, and lifestyle interventions in patient counseling for mental health and neurocognitive disorders. Some of the discussion topics will include the gut-brain axis, key pathways and mechanisms, GI hormones, neurotransmitters, and microbes, how diet, antibiotics, and stress can disrupt our microbiome. We'll define dysbiosis and link it to neurocognitive disorders. And finally, touch on how probiotics, dietary, and lifestyle interventions can support the microbiome and our mental health and cognitive function. First, what comes to mind when you think of the gut-brain connection? Try to think of one word or phrase. In class, I'll pull up a QR code that students can connect to on their phones so we can put up a live word cloud of everyone's baseline. Next, I'm going to play a video that introduces the gut-brain connection. It's a short video, about 1.5 minutes. The audio is not supported in this screen share, so I'll save this video for class. The gut microbiome is a diverse symbiotic community of bacteria, fungi, viruses, and protists in the human intestinal tract. It contains some 2,000 bacterial species and 150 times more genes than the human genome. The microbial mass weighs approximately 2 to 3 pounds, and it develops after birth, influenced by diet, age, and the environment. The gut microbiome has many functions, including to digest complex carbohydrates like dietary fiber, helps with immune system development and maturation, helps support energy homeostasis and metabolism. Microbes produce essential vitamins like B and K, and they conjugate bile acids to break down fat. The gut-brain axis is a bidirectional communication system via neuroendocrine, immune, and microbial metabolite pathways. The neuroendocrine pathways include the enteric nervous system, the vagus nerve, and the hypothalamic pituitary adrenal axis. Immune components include cytokines and microglia. And microbial metabolites include hormones like serotonin, GABA, and dopamine, and a metabolite called short-chain fatty acids. And we'll talk through each of these different components on the next few slides. I like the way this picture brings together all the components of the axis, and I'll use it as a reference point as we talk through all the different parts, so you can see how they all work to connect the gut and the brain. First, the enteric nervous system and the vagus nerve. The enteric nervous system is often referred to as the gut brain or the second brain. It's a nerve fiber network within the intestinal submucosa. And the vagus nerve is cranial nerve 10 from the brainstem. It's a high-speed bidirectional pathway that links the enteric nervous system to the central nervous system. Interestingly, 90% of impulses transmitted across the vagus nerve go from the intestines to the brain. It's kind of like the interstate freeway. The HPA axis and cortisol. The HPA axis is our body's stress response system. In the face of a stressor, the hypothalamus produces corticotropin-releasing hormone, which stimulates the anterior pituitary gland to release adrenocorticotropic hormone, which stimulates the adrenal glands to produce the stress hormone cortisol. Cortisol then triggers immune secretion of pro-inflammatory cytokines. It's important to note that changes in the gut microbiome can also activate the HPA axis, so they definitely play a role as a stressor. Cytokines and microglia. Pro-inflammatory cytokines are intestinal immune cell signaling substances released into the bloodstream. They form a feedback loop with the HPA axis and have the ability to cross the blood-brain barrier. Microglia are the primary innate immune cells of our central nervous system. They perform neurogenesis, maintain the integrity of the blood-brain barrier, and also help with phagocytosis, kind of like the cleanup crew in the brain. The gut microbiome also signals microglia, and they do this via short-chain fatty acid release. Short-chain fatty acids are gut microbial metabolites produced by anaerobic fermentation of indigestible complex carbohydrates like dietary fiber. They have many functions. Some of them include regulating the synthesis of GI hormones like GLP-1 and PYY. Short-chain fatty acids also inhibit pathogenic bacteria in our gut and help to restore intestinal barrier integrity. One important short-chain fatty acid is called butyrate. Butyrate serves as the primary energy source for colon cells, and research has shown it has a role in enhancing learning and memory, reducing depressive-like behaviors, and increasing sociability in mouse models. Neuroendocrine messengers. There are many GI hormones. Enteroendocrine cells produce GLP-1 and PYY, and these stimulate insulin secretion and regulate satiety, or feeling of fullness, and our mood. Ghrelin is a hunger signaling hormone and also helps modulate mood and cognitive function. White adipose tissue produces leptin, which helps with energy homeostasis and immune function. Most of us are familiar with serotonin. This is the happiness hormone that regulates GI tract activity, mood, pain, and sleep. And the gut is an important site of production of serotonin. 95% is produced by enterochromaffin cells in the intestinal lining. Serotonin deficiency in the central nervous system has been linked to sadness, apathy, and anxiety. So the microbes themselves also produce metabolites that are transmitted to our central nervous system as hormones and neurotransmitters. Some examples of this include Enterococcus and Candida species, which produce serotonin. Bacillus species produce dopamine, the primary reward hormone. And Bifidobacterium and Lactobacillus species produce the main inhibitory neurotransmitter, GABA. It's time for a gut check. Name 3 hormones produced in the gut that affect the brain. I'll give you a moment to think about it. We covered quite a few, but some examples are serotonin, GLP-1, and ghrelin. Which gut microbes produce GABA? Bifidobacterium and Lactobacillus species. Which short-chain fatty acid may help to enhance memory and reduce depressive-like behaviors? Butyrate. Think, pair, share. Tina is a 32-year-old with irritable bowel syndrome, anxiety, poor sleep, frequent antibiotic use, a high processed food diet, and a PHQ-9 of 17. Turn to a partner to think about the following questions. What gut-related factors could be worsening her mental health? What would you prioritize in your patient counseling? In class, I'll have students talk with each other for 2 to 3 minutes, and then I'll pull up another interactive platform that they can connect to with their phones via QR code so that we can see in real time different people's thoughts about these questions. I expect this activity to take around 5 minutes. Dysbiosis. Dysbiosis is defined as changes in the composition and function of the gut microbiome. We see an increase in pathogenic bacteria and overall loss of microbial diversity. So the picture on the right shows the diverse, healthy gut with lots of different types of bacteria. But in the face of dysbiosis, we only have 1 or 2 strains that are taking over. And this can dramatically impact the health of our GI system and other systems at large too. Notably, dysbiosis was recognized as one of the 12 hallmarks of aging. There are many things that can disrupt our gut microbiome. Today we'll be talking about ultra-processed foods, broad-spectrum antibiotics, and the role of stress and sleep deprivation. Ultra-processed foods are defined as industrial formulations of food substances that contain relatively no nutritional value. Some examples include sodas, snacks, confectionery products, ice cream, and pastries, and also pre-prepared dishes. These foods have high levels of refined sugars, saturated fats, and synthetic additives like artificial sweeteners, preservatives, and emulsifiers, and this can trigger gut dysbiosis, microbial imbalance, and intestinal inflammation. It's important to note that ultra-processed foods make up over 50% of the caloric daily intake in the US. Ultra-processed foods are often more accessible and cheaper than fresh or unprocessed food options and are also very heavily marketed. So this study was an interesting way to look at the impact of food emulsifiers on the human gut microbiota. So researchers made a simulator of the human intestinal system in the lab called the Mucosal Simulator of Human Intestinal Microbial Ecosystem. And this contained all the different parts of the GI tract and the chemicals that it would contain closely. And then it was inoculated with fecal human microbiota so that they could see the impact of these chemicals on the microbiome themselves. So food emulsifiers P80 and CMC were applied chronically over 7 days to mimic dietary exposure to the microbiota. And the researchers found that these emulsifiers directly affect composition and gene expression of human gut microbiota. Specifically, it increased levels of bioactive flagellin, leading to inflammatory immune response. Notably, these changes occur independently of a living host, which suggests the direct impact of food emulsifiers on the human gut microbiota. So, very interesting experiment and a way for us to look at the role of the chemicals of ultra-processed foods on our gut microbiome. Broad-spectrum antibiotics eliminate pathogenic and beneficial bacteria, leading to colonization by antibiotic-resistant pathogens. This causes a risk of chronic dysbiosis in the gut microbial community. It's important to note, prolonged antibiotic use in infancy and early childhood may impact early immune system development. Stress and sleep. Chronic stress leads to a dysfunctional HPA axis and the production of excess cortisol. It causes a significant decrease in bacterial species Lactobacillus and Bifidobacterium. Remember that these two species are the ones that produced GABA, the main inhibitory neurotransmitter. GABA is associated with a reduction in stress and anxiety symptoms and reduced inflammation. So a decrease in these species promotes a positive feedback loop of that dysfunctional HPA axis along with excess cortisol. So you have a positive stress release. Also, it's been associated chronic stress has been associated with reduced integrity of the intestinal barrier. So the gut microbiome has its own circadian rhythm. It's sensitive to phase shifting like jet lag, shift work, and interrupted sleep patterns. It's been found that sleep deprivation less than 6 hours a night can contribute to dysbiosis and intestinal barrier disruption. Leaky gut syndrome is defined as a disruption in gut barrier epithelium, which leads to increased intestinal permeability. It involves the translocation of gram-negative lipopolysaccharides and the production of pro-inflammatory cytokines and microglial activation. So the picture on the right demonstrates the difference between a normal gut with the epithelial cells lined up with their normal tight junction. This creates a barrier that prevents the pathogens in our GI tract from entering our bloodstream. However, on the right, when you have a leaky gut and there's inflammation, there's little gaps where the bacteria and metabolites and pathogens can pass through and enter the bloodstream. And this can send signals to the immune system at large that there are inflammatory components. So how does this connect to mental health? One interesting example is in the case of tryptophan metabolism. So serotonin is produced from the precursor amino acid tryptophan. And in a normal, healthy gut microbiome, when all systems are going as they should, tryptophan will be metabolized into serotonin. However, in the face of a stressor like dysbiosis or leaky gut syndrome, you'll get an excess of inflammatory markers, those pro-inflammatory cytokines, and this can actually switch the tryptophan metabolism pathway to a different pathway called the kynurenine pathway. So unfortunately, this leads to the production of neurotoxic metabolites and a decrease in serotonin. Serotonin deficiency in the central nervous system has been linked to depression and anxiety symptoms. So a really kind of interesting way to look at how our gut microbiome can directly affect our mental health in this example. Systemic inflammation and the brain. Over time, chronic gut dysbiosis leads to increased permeability of the blood-brain barrier, also promoting cytokine accumulation, which kind of chronically activates microglia. Chronic microglial activation is associated with persistent low-grade neuroinflammation. And these two features are present across neurodegenerative diseases like Alzheimer's and Parkinson's. Additionally, dysfunctional microglial phagocytosis leads to neuronal loss and cognitive decline. So a whole host of issues triggered just from dysfunctions in our gut. Pretty scary. So this is an FYI slide. I wanted to put together all of the research, kind of the highlights that have linked the imbalances in our gut microbiome to the development of neurocognitive disorders. And this is emerging evidence. There's still a lot of studies happening, but a kind of underlying common theme is that the disruptions, especially chronically, that lead to neuroinflammation can cause these symptoms to worsen in these different conditions. And also, ultra-processed foods can play a role in worsening symptoms as well. But if you're interested, I've linked all the different sources here so that you can look into the research yourself. Gut microbiome defenders. So we are able to support our microbiome through dietary approaches and probiotics, judicious use of antibiotics, and exercise, stress management, and sleep hygiene. First, high fiber foods. These types of foods promote short-chain fatty acid production, like butyrate, by our gut microbes. Remember that the gut microbes will, aerobic microbes will ferment the indigestible fibers, and they use that as energy. So this helps to reduce neuroinflammation and restore the integrity of intestinal and blood-brain barrier walls. Some examples of high fiber foods are flaxseed, popcorn, oats, and almonds. Fermented foods contain probiotics, which may increase microbial diversity. Researchers found this may also improve mood, anxiety, and cognition via GABA and serotonin modulation. So 3 different studies showed an increased abundance of lactobacillus bacteria reported after a 2-week administration of kefir, fermented soy, and yogurt. These were different studies that looked at the different fermented food products. Some other fermented foods include sourdough bread, cheese, and miso. The Mediterranean diet is well known for being rich in omega-3 fatty acids, fiber, and polyphenols, and research has linked it to a lower risk of depression and cognitive decline. One interesting study that I'll bring up is called the SMILES trial from 2017. This was a randomized controlled trial involving adults with major depressive disorder. Following a 12-week Mediterranean diet where participants were randomized to either a dietary intervention group or social support control group, the researchers saw significant improvements in mood and reduced anxiety levels in adults with major depressive disorder. And notably, 32% achieved remission of symptoms after the 12-week Mediterranean diet. So, a pretty powerful example of how dietary intervention could be used as a treatment strategy for major depressive disorder. The Mediterranean diet focuses on fruits, veggies, whole grains, and extra virgin olive oil, also fish, seafood, nuts, and legumes, and it limits the intake of red meat, sweets, poultry, low-fat, dairy, and eggs. The ketogenic diet is a high-fat, high-protein diet with reduced carbohydrates, typically only 5 to 10% of dietary intake. It's been found to have neuroprotective benefits, enhanced mitochondrial function, and reduces oxidative stress. Keto is clinically advised for drug-resistant epilepsy, and there's also been emerging evidence for Alzheimer's, Parkinson's, and bipolar mood disorders. Probiotics and prebiotics. Probiotics are live microorganisms with a health benefit for the host. So some of the old school ones are the Lactobacillus and Bifidobacterium species, but you've also got some next generation probiotics like Akkermansia muciniphila. And this is a very interesting probiotic because research has found that it's linked to improvements in insulin sensitivity as well as lipid metabolism. So potentially could be used as an intervention for folks who have type 2 diabetes. Prebiotics are the food used by the microorganisms. So these non-digestible substances that promote growth of the beneficial bacteria. So there's quite an interesting category of prebiotic foods. You see like bananas are pretty accessible, but I don't know necessarily where you would find wheat bran or dandelion greens, but you've got a lot of different options of ways to feed your gut microbiome. Research associates probiotic supplementation with symptomatic improvement in major depressive disorder and Alzheimer's. However, not all probiotics are considered equal. So if you go to over-the-counter probiotics as an option, you need to make sure that you're getting a source that's high quality and it's from a reputable production process. So oftentimes the research shows it's best to consider a food-first approach and to tailor supplements to individuals. Antibiotic stewardship. As we know, it's very important to practice judicious use of antibiotics. We want to avoid overprescribing, minimize broad spectrum exposure, so antibiotics such as clindamycin and fluoroquinolones. And we also want to use caution with prolonged or high dose courses, especially for infants and young children, as this could shift their microbial diversity, which may impact immune development. There are also some powerful lifestyle modifiers that can support our gut microbiome and our mental and cognitive health. So these include exercise. Regular physical exercise is a powerful way to increase our microbial diversity, reduce stress, and systemic inflammation. We also want to practice stress management. Exercise is a great way to do this, and meditation and cognitive behavioral therapy can also regulate our HPA axis. We would like to practice sleep hygiene, ideally greater than 6 hours a night, and you want to establish consistent sleep routines. And finally, smoking cessation is another lifestyle modifier that has a powerful effect on increasing our microbial diversity. One source said quitting smoking is the best solution to treatment of disease related to altered gut microbiome composition. So we've learned a lot of different ways that the gut microbiome is impacted and ways that we can protect it. So we'll apply these now to a clinical decision-making quiz or application. In class, I'm going to do this as an interactive QR-linked multiple choice quiz so people can see it in real time. But for the purpose of this podcast, I'll just kind of go through the answers. A 45-year-old assigned male at birth with chronic stress, insomnia, and brain fog asks how stress may impact his gut and brain. What is the most evidence-based explanation? So you won't be able to see all the answer choices, but the answer is B, stress-induced gut permeability allows bacterial metabolites into the bloodstream, causing neuroinflammation. The rationale is chronic stress can induce leaky gut, allowing lipopolysaccharides from gram-negative bacteria to enter systemic circulation, activating immune responses, and disrupting the blood-brain barrier, all contributing to neuroinflammation. So bringing back that idea of a leaky gut. A 9-year-old child with drug-resistant epilepsy is placed on a ketogenic diet. Besides ketone production, what additional mechanisms support the gut-brain axis? So again, classmates would participate and see 5 answer choices, but for the purposes of this PowerPoint, I'll just show the answer. The answer would be neuroprotective benefits, enhanced mitochondrial function, reduced oxidative stress. Keto is a high-fat, high-protein diet with reduced carbohydrates, 5 to 10%. Emerging evidence suggests neuroprotective benefits for Alzheimer's, Parkinson's, and bipolar mood disorders. A 25-year-old assigned female at birth presents with new onset anxiety, poor sleep, and bloating 2 weeks after finishing ciprofloxacin for a UTI. She reports eating mostly processed foods. What is the most evidence-based recommendation to support her microbiota recovery? The answer would be adopt a Mediterranean-style diet rich in fiber and fermented foods. Antibiotics can cause long-term dysbiosis, impacting the gut-brain axis, and may increase the risk of anxiety and depression. A Mediterranean diet rich in fiber and fermented foods supports short-chain fatty acid production and microbial diversity. So, key takeaways. The gut-brain axis is a complex system connected through neuroendocrine, immune, and microbial pathways. Gut microbiome dysbiosis can directly influence mood and cognition. Ultra-processed foods and antibiotics reduce microbial diversity and impact brain health. And we can help our patients with personalized dietary and lifestyle interventions to support mental health and lifelong cognitive function. So, as a final activity with the class, name one way you'll use gut-brain principles in your future clinical encounters. I'll pull up a similar QR-linked forum so we can see how everyone is going to apply this to future patient care. Here are my references. And I found some really helpful additional resources from the School of Medicine and Public Health at University of Wisconsin-Madison. They created a variety of different patient handouts that I think do a really good job of summarizing, promoting a healthy microbiome, also explaining prebiotics and probiotics, frequently asked questions, and also how to follow different diets like a Mediterranean diet. So feel free to check those out. And we have time for questions. So thank you so much for listening to my presentation, and I appreciate any feedback you have.